Greetings to you, dear students. So this is a complete cycle of Krebs cycle. Already in one video, I have explained all steps involved in the Krebs cycle. But in this video, little depth clearly I shall explain about the Krebs cycle. You could watch carefully and learn the cycle as it is and follow the explanation. So already in glycolysis, pyruvic acid is formed or pyruvate, two molecules. So again those pyruvate molecules enters into Krebs cycle. Krebs cycle occurs in mitochondrial matrix. So firstly, this pyruvic acid is converted into acetyl CoA. So pyruvic acid combines with CoA and with the help of pyruvic dehydrogenase enzyme, it will be converted into acetyl CoA plus CO2. So here this mechanism is known as beta oxidation. Through beta oxidation, pyruvic acid is converted into acetyl CoA plus CO2. Here NADH2 is released and CoA is involved. So our cycle, Krebs cycle, starts with acetyl CoA. Here what happened? This acetyl CoA combines with this oxaloacetic acid, 4 carbons containing oxaloacetic acid, and it forms citric acid. So acetyl CoA 2 carbons, oxaloacetic acid 4 carbons, 4 plus 2 6. So here first stable product citric acid is formed. That citric acid contains 6 carbons. Next, citric acid is converted into cis-aconitic acid. In this conversion, aconitase enzyme is helpful. Here one molecule of water is liberated or released from the reaction. That is called dehydration. Then this cis-aconitic acid is converted into isocitric acid. Here the enzyme is aconitase is involved. Here one molecule of water is added in the reaction, that is called hydration. Later isocitric acid is converted into oxalosuccinic acid. Here the enzyme is isocitric dehydrogenase is involved, and from this reaction NADH2 is liberated. So these oxalosuccinic acid also 6 carbon containing compound. This oxalosuccinic acid again converted into alpha ketoglutaric acid. This is 5 carbon compound. In this reaction, one molecule of carbon dioxide is liberated. This is called decarboxylation. Here an enzyme is used, oxalosuccinic decarboxylase. And again alpha ketoglutaric acid is converted into succinyl CoA. Succinyl CoA also 5 carbon containing compound. In this reaction, alpha ketoglutaric acid dehydrogenase is involved enzyme. Here also NADH2 is liberated. Then succinyl CoA is converted into succinic acid or succinate. Succinate or succinic acid is 4 carbon compound. Why? Because in this reaction, one carbon dioxide molecule is liberated, it goes out. So 4 carbons compound is formed. The enzyme is called succinic acid thiokinase. And in this reaction, one ADP is involved and ATP is liberated. This succinic acid again converted into fumaric acid. Here the enzyme is succinic acid dehydrogenase is involved. In this reaction also, FADH2 is liberated. Later, this fumaric acid converts into malic acid, 4 carbon compound containing malic acid. Here the enzyme is used, that is called fumarase, and one water molecule is involved in this reaction. So that is called hydration. Then finally, 4 carbons malic acid is converted into oxaloacetic acid, 4 carbons. The enzyme is malic acid dehydrogenase. Here also NADH2 is liberated. Then vice versa. Again this 4 carbon containing OAA, oxaloacetic acid. Again it combines with acetyl CoA and it forms citric acid, the first compound, and the cycle will be repeated. So continuously the cycle will be repeated. So these all NADH2 and FADH2 finally enters into electron transport chain to produce final ATPs, adenosine triphosphate. Electron transport chain is third phase. So in respiration, glycolysis first phase, Krebs cycle second phase, and electron transport chain third phase are involved. Finally ATPs are produced. So this is the final schematic cyclic representation of Krebs cycle. So dear students, learn well and score the marks well. Thank you. Signing off, Mr. Karthikeyan here.